does a microscope work and why does it create a magnified image? What is the greatest magnification that an optical microscope can make, and what sets the limit to it? Why is it impossible to see atoms through an optical microscope? We will answer these questions in this video. The simplest kind of microscope is a tube with two short-distance collecting lenses at its ends, an objective and an eyepiece. An objective is called so because it is directed at an object that is being observed. An eyepiece is used for looking into it with an eye. An object is placed onto a stage and is illuminated so that the image is bright enough. Let us draw an objective lens and mark its foci. An object is placed in front of a focus of the objective in the nearby space, in which case the object's image is going to be actual, reversed, and greatly magnified. An eyepiece is an ordinary magnifying glass. But here we use it to observe not the object itself, but its image, obtained by means of the objective. Let us draw convenient rays and reproduce the image we can see through the eyepiece. The image moves to the distinct vision distance and gets magnified. The maximum magnification a microscope can produce equals objective magnification multiplied by eyepiece magnification. Let us make a model of a microscope. We put a collecting lens onto the optical bench. This will be our objective. We place a brightly illuminated object behind the focus of the lens and project its reversed and magnified image onto a screen. We'll be observing this image through another collecting lens, which will be performing the function of an eyepiece in our microscope. If we remove the screen, the image will become much brighter. The basic type of microscope that we have just considered was invented in the beginning of the 17th century. Its invention is attributed to different individuals. This type of microscope cannot provide really strong magnification due to great optical distortions. A German scientist, Ernst Abbe, suggested significantly more sophisticated types of microscopes in the second half of the 19th century. The microscopes by Carl Zeiss Jena, made according to Abbe's calculations, were the first to allow reaching the optical resolution limit and observing objects whose size is comparable with the optical wavelength. Bacteria are clearly observable through such a microscope, but viruses, whose size is much smaller than 1 micron, are impossible to see. It is true for atoms all the more. You need an electronic microscope instead of an optical one if you want to see those.